The measure of intelligence is the ability to change. Hi everyone, Dr. Clark here again, and we're going to continue talking about the principles of science by talking about the theories that unify, or the theories that nearly all of biology are based on. So there are a group of theories that no matter what field you're talking about, these theories play a very important role in each one of those fields. It's kind of like the things that, the ideas that unify biology. You can think of it that way. And so, well, and when I say ideas, I mean these are supported with evidence for hundreds of years at many times. So these aren't theories in the sense of guesses. And we're going to cover that first. What is a theory? So a theory to a scientist means something very different than a theory to the general public. A scientist's theory means that something is certain. It represents a explanation for a natural phenomena that is certain, that we're basing all this evidence on, and it has not been rejected. Tons of experiments have been done on it. Never has been rejected as the explanation. Therefore, it's tentatively accepted. But that tentative acceptance of the original hypothesis or hypotheses has been elevated to a theory, so we assume that it is certain, okay, and that there's a level of certainty to that theory. That doesn't mean that that theory can't change. Lots of theories change. That's what separates a theory from a law. Laws should not change, because laws are 100% certainty. Okay? A theory is, right now, in this moment, based on all the evidence that has been presented over the last 100 years, 200 years, 300 years, this is the explanation for the natural phenomena. To the general public, very different. The general public, a theory implies actually a lack of knowledge or a guess. You'll often hear people say this: I have a theory on that, okay, just in general conversation. And to a scientist, when we hear that, at least most scientists, we cringe, because we know that they're using the term theory to imply that they have a guess, to imply that they don't have knowledge on it, but they got kind of this wacky idea about something. So the two are very, very different. Literally, to a scientist, it is certainty. To the general public, it is a guess or a lack of knowledge. And that is where those terms really get confused with people when they hear things like the cell theory, or the theory of disease, or the germ theory, or theory of evolution. A lot of the general public say, well, those are just guesses. I mean, it's just a theory. I hear that quite often. It's just a theory. Well, to scientists, it's not just a theory. It is the theory. It is the explanation for that natural phenomena. Okay? It's not 100 different theories or guesses, according to the general public, about certain subject material. It is the single theory that explains that topic. It's the single theory that has been tested numerous times and never been rejected, that explains that natural phenomena. So that is where that term theory really gets confused with the general public. And part of that really starts with, education at a young age. We allow that term theory to be used as a guess, but it's not a guess, and we should stop using it as a guess when we're talking about scientific material. All right, so scientists' acceptance of a theory is provisional. Of course, that's what I already just stated. Look, everything in biology for sure, okay, should be recognized as tentative. For that matter, all theories should be recognized as tentative. Now, the theory of friction, is it likely to be rejected? No. But it still should be a tentative explanation for that process. Theory of evolution, is it likely to be rejected? No. But it's still tentative. And you'll see that as we progress, I'm going to talk about Mendelian theory, the theory of genetics. Okay? And we're going to talk about the 5 hypotheses that Mendel had to develop that theory. And you can see that today, 2, maybe 3 are now rejected. Okay? But during Mendel's time, those hypotheses were supported with his data. And when I say rejected, the only reason they're rejected is because now we can do genetic analysis. We now know more about genes and DNA and loci. Mendel didn't know there was such thing. Mendel was making mathematical explanations for a natural phenomena of how things are inherited. Now we're actually looking at the material that's actually being inherited. So Mendel, one of his hypotheses was that genes don't interact with other genes. But we now know, due to lots of individuals' work, but mainly Barbara McClintock, her work shows that genes actually have the ability to jump around, move places on a chromosome, and we know that gene-by-gene interaction happens all the time. Mendel couldn't figure this out because he didn't even know genes existed. So yes, we had to reevaluate the theory of genetics, Mendelian theory of genetics. We had to reevaluate it with today's knowledge, with today's technology. So that's why theories are provisional, because as we combine and add more evidence, you might have to adjust parts of that explanation. Okay. That kind of brings me to this, the premise of guesses. Okay, I know that a lot of students are taught that hypotheses are educated guesses, that theories are guesses. Okay, science is not guesswork. Science has nothing to do with guessing. Science has to do with an explanation for a natural phenomena. Now, if your explanation is wonky or not testable or just ridiculous, then you're not doing science, because that violates the scientific method. The scientific method means that you have to have an explanation for a natural phenomena, but that explanation has to be testable. That testability is what makes science not trial and error. Right? That testability means that we don't just come in with an idea and test every single thing possible and then, present our results. No. We talked about this a little bit before. We use inductive and deductive reasoning. If deductive reasoning says there's a principle out there, there's a theory out there, there's a law out there that would violate one of our experimental variables or violate one of our hypotheses, then why test it? Okay, it's not trial and error. It's using deductive reasoning and inductive reasoning at the same time. Okay, that's where the judgment and intuition comes in. Okay, you're judging whether or not you even need to test this explanation, this hypothesis, because you already know that there's a law that states that that's not possible. Okay? Now, that doesn't mean that you can't test it. But again, it's a judgment call, and there's intuition, and it's based on prior knowledge of scientific principles. It's not based on prior guesses. It's not based on what your grandma told you. It's not based on the political views at the time. It's based on the prior science. All that being said, science has its limitations, though. Okay? And I want to make sure that it's clear that this is my opinion. This is Dr. Clark's opinion. Okay? There are a lot of scientists that don't agree with this, and I respect their opinion, but this is my opinion. And that is, science is limited to the ability to measure something, observe and measure something. Now, that doesn't mean that your imagination isn't important. Einstein says, a scientist's imagination is far more important than the scientist's knowledge. And in part, I agree with Einstein's views that if you can't think of the possibilities that are out there, the possibilities of things that you can't see, the minute microorganisms, the energy you can't see, the wavelengths of energy you can't see. If you can't think about the possibilities, then you're limiting yourself. But again, science has limitations. Science is limited to those things that we can observe and measure. So, in many parts, when we make explanations about things that we don't have a technique to measure yet, that's really not science. Okay? That's guesswork, and it's also often called pseudoscience. So if you're making a guess about how something occurs, but we don't have any physical way to measure that or observe it, then it really is a guess until you do have the technology to measure it and observe it. Okay? So that brings me to things like supernatural events, religious phenomena, other things. These are beyond the scope of science right now. Okay? You can't observe them. Okay? Even though individuals say they've observed them, you can't repeat that observation at a consistent time and matter. So you can't call up that supernatural event over and over and over again and measure it and test it. Okay? That's not possible right now. Okay? Same thing with religious phenomena. So in situations like this, it is my opinion and my opinion alone, although I'm sure some other scientists agree with me, that scientists should not be discussing supernatural events, religious phenomena, etc., because that's not science. Now, if you're discussing it because it's your personal opinion and you're not discussing the validity of it as science, then I'm okay with it. You can discuss whatever you want, as your personal opinion. But if you're talking about it from the validity of science, then that's not science either. You're practicing pseudoscience because you don't have a way to measure this. You don't have a way to observe it, etc. Okay? All right, so that brings me to the 4 theories. Okay, remember, theories are not guesses. These are supported evidence for natural phenomena, and they are the accepted explanation for that natural phenomena. These theories are the cell theory, the gene theory, the theory of heredity, and the theory of evolution. All right, so let's start with the cell theory. Start with Robert Hooke. Robert Hooke, interesting character and often forgot, to tell you the truth. The only thing people really remember about Hooke is maybe that he had a big ego, that him and Newton didn't get along, and then some people remember that Hooke is the individual that first described cells. Okay, so let's talk a little bit about Robert Hooke. Robert Hooke used and figured out that you could stack glass. Okay, to make and magnify an image. So he made a microscope. Again, he's not the first person to make a microscope. There were people that had already made telescopes and other things, okay? But he did make his own microscope, and he examined cork, okay? Cork is a substance right underneath the bark of a tree, okay? And it's typically a bunch of dead cells that provide protection from insects that are burrowing into the tree, or sometimes it provides an extra protection from fire or disease and these kind of things. So he examined cork, and in examining cork, he saw rectangles. And the story goes that these rectangles, most of the stories go that these rectangles reminded him of the areas, the housing units that monks used to live in. And those housing units were called cells. So that's where he got the term cell from, is these little monk-inhabited regions or monk homes that are rectangular. And so that's where the term cell came from. Now, Robert Hooke, the reason why he sometimes gets a bad rap is Robert Hooke really, he wasn't... in today's society, you would call him very nosy, or he didn't know about kind of personal boundaries or scientific boundaries for that matter. If he had an idea, if someone else had an idea and he read their material and he thought that their idea was worthy of testing, or that their idea was not a good explanation, etc., he would call him out on it, or he'd do his own test. And there's quite a bit of literature, quite a bit of evidence that suggests that Robert Hooke actually is the very first person to write about gravity. Even though Newton gets credit for the kind of discovery of gravity, Robert Hooke, it looks like, from the evidence of his journals and everything, he knew about it before Newton described it, and Newton may have stole Robert Hooke's ideas on gravity. But because Hooke was kind of rude, and he wasn't a very nice guy, neither was Newton, but Hooke was often pushed away from different science fields, chemistry, physics, things like that. People didn't like to interact with him. So that's kind of the story around Hooke. Again, he's the individual that described the first cell. Right? So he discovered cells, and he coined the term cell. Antoine van Leeuwenhoek, okay, in 1670, a little bit after Robert Hooke, he started using the idea of microscopes and mirrors and things like that, and he started creating all kinds of microscopes, different variety of microscopes. I think the count that's often given is over 250 microscopes. Antoine van Leeuwenhoek developed, engineered over 250 microscopes. Well, his work allowed him, because he made better microscopes than Hooke did, his work allowed him to examine other things like pond water and other things, and he discovered single-celled life. Okay, most likely he discovered bacteria or protozoans, or both. Some of his sketches look a lot like protozoans, some look a lot like bacteria, but nonetheless, they didn't know what either one was, but they knew it was single-celled life. Then came Malthus Schleiden and Theodor Schwann, and much later, microscopes had advanced quite a bit, but these two individuals started examining all kinds of organisms. Schleiden was a plant guy, Schwann was an animal guy, and they just started looking at everything. They looked at cells of different types of plants, their roots, their flowers, the pollen, the ova, and then Schwann was looking at sperm and egg and blood and skin and hair, and they're just looking at everything. And they came up with a kind of tenet of the cell theory, which is that all organisms are comprised of cells. And the second tenet, okay, now all of these individuals kind of add to this cell theory, but there are three tenets or three parts to the cell theory: that all organisms are composed of cells, that cells are the basic unit of life. So in other words, you can have single cell life. Okay? And then the third tenet, which is that all cells come from pre-existing cells. Okay? Or cells are not created, rather they are multiplied from other cells. Okay? And that's a big one, because this third tenet of the cell theory was a way to get rid of this spontaneous generation idea that things poofed into existence. Okay? And one of the individuals that really added to that, really kind of discovered that third tenet of the cell theory, was this man. Okay? And this is Louis Pasteur. Louis Pasteur, some of you have probably heard of his name because you drink pasteurized milk, and he came up with that process so you have the ability to drink milk for a longer period of time through the pasteurization process. But Louis Pasteur designed an experiment where he was taking flasks like these, sometimes called the swan neck flask, also called Pasteur's glassware. Okay, so he developed these flasks. He would take nutrient broth, which is nutrients like nitrogen and phosphorus, potassium, things like that, that's mixed into water, that allows for bacteria to grow. And we'll get to what they're using those different components for as we progress, but it allows for bacteria to grow. Bacteria, fungi would grow in these flasks, probably viruses also. But again, none of them had really been discovered or described to that point. Some of the fungus were, so you would know that it was fungus. But at any rate, it would make that flask cloudy. And you could see, and if you threw it under a microscope, you could see that there was life in there. And then the other ones, they had nothing. And so what he would do is he'd boil these broth. Okay, he'd break the tops off of some of them, allow the air, so breaking the top off would allow things from the air to come into the flask, and then having these swan necks would not allow air and particles to come into the flask. And he'd sit them on his lab bench, and he sat them for days, weeks, months, years, and he did observations. And he did this for a long period of time until he came up with that part of that tenet, or that third tenet to the cell theory, that all cells come from pre-existing cells, that there is no spontaneous generation, that the material that's in the flask is coming from microscopic organisms that are floating around in the air that you can't see, that are not visible to the naked eye. And that's where the cells are coming from. That's where moldy bread comes from. That's where rotten meat comes from, are things that you cannot see with the naked eye that still exist. And so Pasteur is also credited for, pieces of germ theory, pieces of the theory of disease. So he had a very important role when it came to this, what we now would call microbiology kind of field. All right, so the gene theory. Gene theory has to do with typically two molecules: DNA, or deoxyribonucleic acid, and RNA, ribonucleic acid. And gene theory is really interesting because as we progressed scientifically, a lot of Darwin's suggestions around evolution, Mendel's suggestions around genetics or inheritance, it really came into question, or came into the ability to test, when gene theory was developed. And so now we had an idea because we knew the genetic background and we knew what genes were, we knew what DNA was, RNA, chromosomes, loci, we knew what these things were coding for. So now we could test, was Mendel right? Were his pea experiments good? Okay. Was Darwin right? Did Darwin's finches, the finches on the Galapagos Islands, actually come from the mainland? Are they actually connected? Are humans actually connected to chimpanzees? Are they our closest relative? So once gene theory was developed, we could start diving into a massive amount of science. Okay? Gene theory also showed us that the system is not, I shouldn't say, it's not perfect. Okay? That you can tell that it was pieced together as it developed, as it evolved over time. It was pieced together because DNA itself does not code for your proteins. DNA has to be transcribed or rewritten into RNA, and then RNA can code for your proteins. So there's these molecules and this kind of steps in the process, okay, that is, I guess you could say, more time-consuming, more resource-demanding, and not streamlined at all. Right? And the more steps you have in a process, the more chances you have for mistakes in a process. And so we can tell that these processes, especially converting your genetic code into proteins, it evolved piece by piece, and it's not a streamlined process. And that was great kind of evidence for that evolutionary piece, that it's not it's not perfect. There's a lot of mistakes that happen. There's a lot of unnecessary steps that occur in the process, hinting at, hey, look, this process evolved continuously for the last basically 3 billion years. Guys, so proteins are coded for by RNA strands. Okay? This occurs in a non-organelle or a certain place in the cytoplasm or associated with a nucleus, depending on whether we're talking about prokaryotes or eukaryotes, and it occurs in a system called the ribosome system, or the ribosomal unit. Okay? Ribosomes have 2 units, a large unit and a small unit, and that is where that RNA code is going to be translated into an amino acid chain, which then wraps itself and maybe binds with other amino acid chains to form complex proteins. Okay, we'll get to all that. I know that might be like, wow, what is this dude talking about? Okay, we'll get to it. But just realize that DNA has to be transcribed into RNA. RNA has to be then translated into proteins. Now, the other cool thing about the gene theory, what it allowed us to figure out, is that if we take intestinal cells, we take muscle cells, we take nerve cells, we take, white blood cells, skin cells, whatever it might be, and we take the nucleus and we look at the chromosomes in that nucleus, all of our DNA is in every nucleus of every cell that has a nucleus. Okay, our red blood cells don't have nuclei, but if they have a nucleus, if they have DNA, all of your DNA is inside that nucleus. In case of humans, all 3 billion base pairs is inside that nucleus. But look at this. Okay? The intestinal cells, these microvilli, they look nothing like your muscle fibers. They look nothing like your nerve cells. So it allowed scientists to say, okay, wait, every one of these cells has all your DNA. But why is it that this cell looks so different than this cell, and this cell, and this cell. And your hair looks different than your skin. And your blood looks different than your toenails. Why? And it allowed us to figure out that, yes, you have all your DNA, every bit, in a nucleus, but not all those genes are turned on. Okay? Our cells have the ability to have genes that are turned off or genes that are turned on, and these turn on and off at different times and due to different conditions. Sometimes it's permanent, and it starts in the womb really early on, that cell differentiates, okay, I'm an intestine cell, and now I'm not going to do anything else. Other times, it takes a while, okay? And then maybe hormones change, and that also changes what DNA is turned on in that cell and what DNA is turned off in that cell. And that's the cool thing about gene theory, is we're just barely starting to scratch the surface on the human genome. Okay, we know that we have roughly 3 billion base pairs of DNA, but only 25,000, 30,000 genes. Okay. We now know that gene-by-gene interaction is extremely important. We know that genes can jump around on chromosomes. We know that the environment can affect your genes. Chemicals that you put in your body can affect your genes. And it's through this gene theory, knowing that connection from DNA, RNA, protein, that we have the ability to start figuring some of this out. All right, theory of heredity. Okay, this is Mendel's baby. Okay, this was Mendel's work. Okay, now in the 1800s, when Mendel was doing his work, we didn't know how offspring had shared characteristics as their parent. We knew they did, but we didn't know how it worked. Okay? And there's lots of excellent explanations. Lamarckian explanations, which was any trait that you acquired in your life, you would pass that on to your offspring. Okay, we know from experiments that were done a little bit later after he came up with this hypothesis that was not true by any means. Darwin had his own hypothesis, which he called gemmules, where little pieces of your body parts would break off, float down to your reproductive organs, and you'd pass that on to your offspring. Okay. There was all kinds of explanations, but Mendel was the individual who started to put it all together. He started to mathematically think about it and look at probability, and he started to come together with what was going on. So Mendel is often considered the father of genetics. Although when we talk about genetics, you'll see that there's a lot of things that Mendel didn't know. And as we go with the genetic field, we now see that Mendelian genetics, it's only maybe 50% of the traits, that an organism has is controlled by Mendelian genetics. Okay, the rest is controlled by a different either gene-by-gene interaction, epistasis, continuous variation, incomplete dominance. There's all kinds of other ways at which your genes interact that determine a trait. But at the time in the 1850s, this was huge. The only thing that Mendel did that I think was wrong is he published his material in an obscure journal that no one read, and it took years before people figured out that Mendel already figured out at least a portion of the theory of heredity. Right? And so the technology advances that we see today, we could have been seeing those in the 80s from a genetics point of view, maybe even in the 70s if Mendel would have published his material in a proper journal. Okay? And we'll look at these because Mendel does have a couple laws associated with Mendelian theory Okay? The law of segregation and the law of incomplete dominance. Okay? And so we're going to look at these. Now, keep in mind, I talked about laws before. Laws in biology are rare, and that's really because, again, biology is always changing, and laws themselves are not supposed to change. But these two laws that are associated with the theory of heredity are mathematical laws. They're based on probability. And so they're a little bit different than, say, a biological law, which has that ability to change over time. And the last one that, unifying theory is evolution. And I talked about this before. Evolution is truly the backbone of biology. Okay? Biology is the study of life. We know that life changes. Okay? Evolution is the change in life over time. And so therefore evolution really has to be that backbone to, life. And Charles Darwin is often associated with evolution, and he should be. Okay? He really came up with 3 ways at which evolution can happen. Okay? Now, it can be argued that other people contributed to these, and they definitely did contribute to them. Okay, like Wallace and a few other individuals definitely contributed to the mechanisms for evolution. But Charles Darwin came up with natural selection with the help of Wallace, sexual selection, probably also with the help of Wallace's research, artificial selection. There's a lot of individuals that were doing this and contributed to that work. Okay, but those are mechanisms by which evolution can occur. Okay. Darwin famously said that, diversity of life is explained by descent with modification. Now, depending on what realm, what individuals you talk about, this has different meanings. To Darwin, it meant that organisms reproduce. Some of the offspring that are part of that reproduction have different characteristics. Some of those characteristics are better suited for the current environment and gives those offspring an advantage to the current environment. Those offspring will then reproduce and pass on that advantage. And as time goes by, that descent, that passing to your children with modification, allows for evolution or allows for adaptation to occur to the environment. Okay? There's a couple other pieces to evolution. Okay? You also have to have isolation. You can't just have adaptation. But we'll talk about that when we get to evolution. And like I said before, genetics was awesome and is welcomed to the evolutionary field. Now, for some evolutionary biologists, it was scary. Because genetics could have removed the entire theory of evolution. If our closest ancestor was not chimpanzees based on genetic evidence, then primate evolution would have been, you'd have to rearrange a lot of primate evolution, and we'd have to look at all those physical features, all those morphologies, the skull, the body parts, etc. We'd have to look at them in a different way if genetics was not supporting that evolutionary connection. Okay? If evolution wasn't supporting the idea that all these finches on the Galapagos Islands, every one of them, okay, there's lots of Galapagos Islands, but all these finches that occur on these different islands, if Darwin suggested that they all came from a single ancestor on the mainland. Genetics now allows us to look at that, and sure enough, when we look at the genetics, and we found a gene called the BMP4 gene, and that's coding for proteins that's placed in the beak of birds, especially these finches, and the more you have, or the earlier this gene turns on, the thicker the bill the bird will develop. Okay? There are lots of other situations that that can change the kind of the shape of the bill, but it just gave more evidence that Darwin in the 1830s, when he was talking about the Galapagos finches and their connection to the mainland, he wasn't wrong. Right? It doesn't mean he was technically right, but currently, at least, the explanation for how the diversity of the Galapagos finches evolved and how it occurred is explained by Darwin's theory of evolution by means of natural selection. So, again, I like to throw it out there that it's a tentative explanation. Now, in this case, that tentative explanation has stood up for 200 years. But in the case of, maybe other explanations that are going on, maybe they've only withstood scrutiny and testing and things like that for 10, 15 years. Okay? So that's why we have to throw that tentative out. Now, that tentativeness starts to go away when you've tested the connection between Galapagos finches and ancestral finches, and you tested them through genetics, you tested them through diet, you've tested them through the ability for them to adapt to changing environments, and you just test and you test and you test and you test, and it's all supported by the theory of evolution by means of natural selection. At that point, that tentative part, it's less tentative. Okay? That doesn't mean that it can't be changed, and it might be changed. Okay? That's why it still remains a theory of evolution and not a law of evolution. Okay, so that brings me to how organisms are classified. Again, that evolutionary part, that's what allows for the diversity of life. Okay? And right now, okay, again, this is the field of biology, so things change. Right now, we break this diversity of life up into 6 major groupings. Okay? That 6 major groupings is typically based on cell structure. Okay? And so when we look at these, we need to talk about this group, the Protista, Plantae, Fungi, and Animalia. Those guys all have nuclei, a true nucleus where the DNA is housed. Okay? They also have some organelles. Now, depending on the cell type, might have different organelles. They might just have a nucleus. They might have all kinds of things, mitochondria, chloroplasts, lysosomes, lots of different types of organelles. Okay? They make up the domain eukarya, okay? Or the eukaryotes. The other 2 kingdoms have their own domain. Okay? The bacteria domain, which is the bacteria kingdom, and the archaea domain, which is the archaea kingdom. Okay? We'll come back and we'll look at each one of these when we start talking about kind of energy, when we talk about evolution, when we talk about how things work. We'll come back and we'll start looking at the differences mainly between prokaryotes, which is over here. Bacteria and archaea are prokaryotes. They do not have a true nucleus, versus eukaryotes, the ones that do have a true nucleus. Okay. All right, so next time we're going to dive in to a little bit on energy, a little bit of chemistry, okay, and kind of lay down the groundwork of how energy works in a biological system. So then we can start talking about some of these systems, and we can start talking about energy flow and what happens when you cut off the energy, or what happens when you add a different stream of energy. Okay, till next time.